So today, as you can see from the title, we're going to be talking about spinocerebellar ataxia and hopefully give you some insights into the aspects of neurodegeneration as they present themselves in Purkinje cells of the cerebellum and neurons within the brainstem. Sort of a brief outline of what we're going to cover this morning. We're going to talk about how the mutant form of ATAXIA1 might be mediating pathogenesis in cerebellar Purkinje cells. We're going to move on to what I think is a rather interesting observation from some RNA-seq study and the identification of a potential protective pathway mediated by the CCK1 receptor. And then finally, we're going to talk about some ASO-mediated therapeutic approaches and their use to assessing pathogenesis, specifically in the medulla in the brainstem. For those of you who are not familiar with SCA1, it's an autosomal dominant form of ataxia. The gene's on the short arm of chromosome 6, linked to the HLA complex. It's typically a late onset disorder, but there are juvenile forms of the disease. And in addition to the ataxia and dysarthria and dysmetria, it is also a lethal disease with lethality being approximately 15 years after onset of symptoms. And we sort of crudely divide the anatomical locations for the two major, if you will, aspects of the phenotype of this disease. The ataxia residing in the pons and the cerebellum, and the lethality due to changes within the brainstem. Specifically, if we look at the cerebellum, this is an individual who's about midway through the course of their disease. You can see dramatic atrophy of the cerebellum as well as the pons here. And then if we move to a section from an individual who succumbed to SCA1, we note a dramatic loss of Purkinje cells from the cerebellar cortex, and those that remain are severely atrophied. In fact, most autopsy material that we've looked at for SCA1, there is essentially a complete lack of Purkinje cells in the cerebellar cortex. So after cloning the gene, one of the first things we tackled was to try to understand pathogenesis specifically in Purkinje cells. And we took this approach for two major reasons. First of all, as we've just gone over, it's a prominent site of pathology in this disease. And as molecular biologists, geneticists, we reasoned that if we could understand the disease process in a specific neuronal type, that would give us a strong experimental platform from which to then expand to other regions of the brain affected by this disease. So our strategy is predicated on a now archaic version of transgenic mouse generation. Simply taking a regulatory region from the endogenous PCP2 L7 gene, hooking it up to various versions of our ATAXIA1 cDNA, and then injecting mouse embryos and generating transgenic mice. This regulatory region is very effective in targeting transgene expression to the cerebellar cortex and specifically to Purkinje cells. If we put it in an expanded version of ATAXIA1 with 82 glutamines, in this overexpression model, this is sort of the general outline of the disease. Initially, the Purkinje cells appear quite normally, and then around 5 weeks of age, we start to see phenotype on the rotarod or balance beam, moving on to 12 weeks where we see a phenotype cage behavior. The important thing to note is that in all of our mouse models, the animals are fully ataxic before we see any loss of Purkinje cells from the cerebellar cortex. So the neurological aspects of this disease are not due to the loss of Purkinje cells per se, but rather a long period of protracted dysfunction. And then eventually in this model, we do see drop-off of Purkinje cells and atrophy of the dendritic tree, very similar to what we showed you in the human material. And again, if we now, in this mouse model, look by Calbindin staining at 1-year-old mice, we can see specifically the atrophy of the dendritic tree and loss of Purkinje cells from the cerebellar cortex. We've also gone on to generate several other models using the same PCP2 promoter. We have a conditional model of the 82-glutamine ATAXIA1. With our colleague Huda Zogby at Baylor, we've generated SCA1 knock-ins, 78 and 154 glutamines into one allele of the endogenous SCA1 locus, as well as an SCA1 knockout mouse. And using these strategies plus various others, we've come to this current picture, if you will, of the ataxin 1 from the standpoint of important functional motifs. The protein we call an 816 amino acid protein. This is based on a glutamine stretch of 30 glutamines. The glutamine track is skewed towards the amino terminus. Downstream from this is a major structured region of the ataxin 1 protein, the so-called AXH domain. It forms what is called an oligonucleotide, or OB motif, as illustrated here in this study from Chen et al. And this is two important aspects of this structure. It forms two interactive surfaces with one of them is with other proteins in the cell, as well as an RNA interaction. And it's also responsible for dimerization of the ataxin 1 protein. So our studies indicate that the endogenous version of ataxin 1 exists in dimers or in larger oligomers. Towards the C-terminus, there's a rich number of functional motifs. There's the nuclear localization sequence. This protein normally shuttles back and forth across the nuclear envelope. If you mutate the NLS on a mutant allele version of ataxin 1, the animals do not get sick. So in order for the mutant protein to have its deleterious effects, it must get into the nucleus. It also interacts with two important groups of cellular proteins. The region around residue 774 to 778 forms an interaction motif with the chaperone class of proteins, the 1433s. This requires phosphorylation at position 776. In addition, there's the UHM motif, which is a common interaction motif among proteins that participate in RNA splicing, and specifically with ataxin 1. In addition to interacting with the transcriptional factor Capicula and other transcriptional factors through its AXH domain, it also interacts with components of the splicing apparatus, notably RBM17, through the phosphorylation site of serine 776 in UHM region. In addition to being important in driving interaction of ataxin 1 with endogenous cellular proteins, phosphorylation of serine 776 also stabilizes the protein and dramatically reduces its clearance by a variety of cell types, including Purkinje cell neurons. So, let's talk about some recent efforts that we have taken in order to try to get at some of the pathogenic changes that occur in cerebellar Purkinje cells, utilizing an RNA-seq strategy. And in this case, we're going to focus on RNA-seq analysis of the cerebellum from two of our ATAXIA models. We've already talked about when we expressed the 82 glutamine protein in cerebellar Purkinje cells. These animals develop a very frank ataxia, as well as atrophy and cell death of Purkinje cells in the cerebellar cortex. In our process of understanding the importance of phosphorylation of serine 776, we generated animals in which we replaced the serine at 776 with an aspartic acid at 776. And this is so a phosphomimetic amino acid residue. And when we place this amino acid residue on a wild-type allele of ataxin 1, we noted that the animals get ataxic, like the 82 glutamine transgenic animals, but the Purkinje cells do not atrophy, and we do not see Purkinje cell loss. So we reasoned that if we could hit RNA-seq on these two transgenic animals, we might be able to get at pathways that are critical for disease progression in the Purkinje cells of the cerebellar cortex. And so this is a study that was published last year on RNA-seq, roughly about 50 million reads per sample, focusing on the 30Q D776 animals, the 82Q animals, as well as wild type, and then animals expressing an unaffected wild type allele of ataxin 1. We had 3 biological replicates at each of these genotypes, and we had 3 ages, 5, 12, and 28 weeks. If you take all of the RNA-seq data and run it through an algorithm called Weighted Gene Co-expression Network Analysis, or WGCNA. And basically what you're doing is sorting all of the genes that you're detecting by RNA-seq into modules or networks that show co-expression with each other across all of these genotypes in all of these ages. And when Christy did this, she got 19 modules, which are identified by different colors for reasons I don't really understand at this point. Not that that's not the only thing I don't understand about WGCNA, but anyway. And one of these modules or networks, designated the magenta module, through subsequent analysis, the data strongly suggests that this is the module that reflects pathways that are directly linked or drive progression in disease in these animals. It's enriched for genes that are highly expressed in Purkinje cells. These genes are downregulated in infected mice, and the degree of regulation decreases even further with disease progression. They're enriched for genes encoding components of G-coupled signaling pathways, which indicates that this might be a particular important cellular process in terms of disease progression. And just to give you an idea of sort of the strategy we've used over the years to try to focus in on things that are critical for pathogenesis, we noted that these genes are enriched for having Capicua binding motifs in their upstream region. And as I said earlier, Capicua and ATAXIN-1 interact via the AXH region of ATAXIN-1. In this study from Kim et al., they noted that if you made several 2 amino acid mutations, you could very dramatically disrupt this interaction by this pull-down assay between Capicua, which has 2 isoforms, long and short, with ATAXIN-1. And so what we did was take our transgenic strategy and generate mice now that have the 82-glutamine ATAXIN-1, but now valine 591 changed to an alanine and serine at 602 changed to an aspartic acid. So in other words, this molecule is no longer able to interact with Capicua. And what's the effect on disease? First of all, you have to control that you're comparing, the 2 things you're comparing have equal levels of expression. So this is true for both mRNA and protein levels. And the bottom line is, despite expressing equally high levels of an 82-glutamine version of ATAXIN-1, these AXH mutant mice do not develop disease, cerebellar disease. So here's the rotarod data at 6, 12, and 20 weeks. The red is the 82-glutamine, and the dark line is our AXH mutant 82-glutamine mice. Going on to pathology, you can see that there's essentially no evidence of Purkinje cell atrophy in the AXH mutant mice. And so the interaction between ATAXIN-1 and Capicua in the nucleus is absolutely critical for the generation of disease in these animals. And I would like to point out that each of these 2 lines, one that has disease and one doesn't, express equally high levels of the mutant RNA with the expanded CAG tract. They each express high levels of the ATAXIN-1 protein, which gets into the nucleus and forms inclusions, but only the animals with the functional AXH domain and interact with Capicua are able to demonstrate disease. Going on and continuing a sort of a second RNA-seq study, we wanted to look at changes, not just changes in expression, but also changes in splicing. And in order to do this, one has to generate much deeper sequence reads. And so this is a study of mice comparing the 82-glutamine with wild-type mice at 9 weeks of age, where we got on average 100 million reads per sample and 5 biological reps per genotype. And we run this data through a couple of programs, algorithms to detect alterations in splicing, one of which Christy used is JunctSeq, and you can see that they identified a whole plethora of altered exon junctions, et cetera, from the sequence data, and 67 of the 321 genes that had evidence of altered RNA splicing, 67 of these overlapped with the magenta module, with a p-value indicated over here. Another RMATS program was also used to look at the data, and a similarly high significant number of genes overlapped with the magenta module. So the magenta genes, which are a gene network that we think is highly important for disease progression, in addition to having changes in expression, these genes also have alterations in RNA splicing. So there seems to be consistent with the interaction of ATAXIN-1 with the transcription factor as well as splicing apparatus, corresponding effects on genes that we think are important for driving pathogenesis. And so here's sort of our current model, if you will, of ATAXIN-1's normal function as well as how this function impacts disease. And the important thing is we're sort of viewing ATAXIN-1 as a coupler of transcription and RNA processing. And I think it's really important for those of you who are doing RNA-seq to appreciate that transcription and RNA processing are linked both temporally and spatially in the nucleus of cells. And you can have changes in splicing by having, changes in transcription and vice versa. You can have changes in transcription rate that are largely due to changes in splicing. So you need to think of this as a more holistic, if you will, mechanism. In an earlier study, we demonstrated that when you have an expanded allele of ATAXIN-1, you drive formation of a complex between ATAXIN-1 and RBM17. And this is sort of outlined over here. So the current concept is that the critical aspect of Copicua is that it brings ATAXIN-1 to sites of transcription on the DNA. ATAXIN-1 itself does not bind directly to DNA. Once at sites of transcription, then it can more readily move back and forth between the transcriptional complex as well as the splicing complex. And phosphorylation at serine 776 and expansion of the polyglutamine tract drives the equilibrium towards the splicing apparatus. And we sort of see the pathogenic mechanism largely be doing to a disruption of the equilibrium between ATAXIN-1's presence in transcriptional complexes as well as in RNA processing complex. And the combination of the two pathways, perhaps with a slightly larger role of changes in splicing, is what now drives changes in gene expression that lead to disease progression. So what about using this same approach to look for protective pathways? And again, we're going back to the 82Q and the 30Q D776 mice and the RNA-seq data that we generated from our first study. Basically, what we hypothesized was that maybe there was a gene or a pathway whose expression was enhanced in the D30 animals, thus blocking disease progression. So at 12 weeks of age, we looked for genes that were significantly different between all of our genotypes. We came up with 130 of these genes by this analysis. 59 of these are higher expressed in the D30 animals, 66 decreased expression in 82, and 32 of the genes had a correspondingly significantly high expression in D30 and significantly low expression in the 82. When we ran these 32 genes through two filters, one of which is highly expressed in Purkinje cells, and the second is showing strong expression by our sequence data, one of the genes that popped up as being very dramatically different in D30 versus the other genotypes was cholecystine kinase, CCK. So cholecystokinin, CCK. It's a long-known neuropeptide that is processed in cells and generates proteolytic fragments that then interact with two receptors, the cholecystokinin 1 and the cholecystokinin 2 receptor. So the next step we did was to take all our mice, and so here's looking at just at the molecular layer thickness, but we looked at other aspects of the phenotype. At 6 weeks versus 1 year of age, you can see the wild type are fairly stable, D30s are stable, and here's the atrophy of Purkinje cells that we see in our 82Q mice. If we cross our D30 mice to CCK knockout mice, we now get a progressive disease. So the absence of CCK on the D30 allele generates now a progressive Purkinje cell atrophy disease. Okay? If we take our D30 mice and cross them to CCK1 receptor knockout mice, so they still have CCK, but they no longer have a CCK1 receptor, we also get progressive disease. Not shown on this slide is when we cross the animals to CCK2 knockout receptor mice. those animals do not get progressive disease. So the model that comes from this is that perhaps that there's an autocrine pathway in Purkinje cells, expression of CCK and its processing then generates a peptide that's secreted and interacts with the CCK1 receptor, giving you a sort of protective activation of a protective pathway. There is an agonist available for the CCK1 receptor. It's a peptoid version of CCK8, A71623. This is its specificity, almost 1,000-fold higher specificity for CCK1 over CCK2. It's also a commercially available agonist for CCK1 receptor. So we then embarked upon several studies where if we could now stimulate the CCK1 receptor using this agonist, can we have an effect on Purkinje cell disease in our various transgenic mice? The first study was to now go to our progressive 30Q CCK minus animals and using an IP pump to now administer this agonist over a period of 35 weeks where we did various phenotypic assessments, as finally ending up with pathology. And I'm just going to show you snippets of the data because of time. Basically, here's the animals that were given the vehicle, and you can see increasingly poor performance by time to cross on a balance beam. And then here's our animals that were given the agonist, and you can see there is no progressive decrease in motor performance. Also not labeled here, you always have to give a presentation with at least one mistake in your slides to kind of give you some evidence that, I don't know, anyway. So here are the two critical bars. Here's a decrease in molecular layer thickness in animals administered the vehicle, the D30s. And then here is the change in absence of a change in decrease in molecular layer thickness in the D30 animals given the agonist. And then here's loss of Purkinje cells from the cerebellar cortex. Again, vehicle-treated D30s as well as agonist-treated agonist-treated D30s. So by both motor performance as well as cerebellar pathology, the agonist was able to protect these animals. We are now more recently moving to, shall we say, more classical versions of SCA models. And basically, we've done both SCA1 now and an SCA2 version from Stefan Pulse Lab using the PCP2 promoter. And here we're only going to be looking at balance beam. Basically, this is an experiment that was designed to give us data in time for an R01 renewal submission that's going to study section in a few weeks. So we are continuing to do more in-depth studies. But basically, what we find in our SCA1 model is, again, back to the balance beam by both foot slips and time to cross. Our agonist is able to protect against increasing motor deficits. And we see a similar protection in our SCA2 model too, again with this agonist. So we're kind of excited that this CCK1 receptor peptide might have some protective value across different diseases affecting Purkinje cells. So I want to end here, hopefully in the next couple of minutes, with some efforts that we've taken to try to understand the pathogenic pathways in a second important region in terms of SCA1. That's the medulla and the brainstem, and specifically as it relates to lethality. Just to point out that our PCP2, or Purkinje cell-specific model, does not show premature lethality, whereas the 154 knock-in mice, where we now have expression throughout the nervous system, we do see a deficit in lifespan, in addition to motor performance in these animals. And at least in human SCA1, it's the idea that the brainstem, and specifically particular regions, most notably in the medulla, is what underlies this lethality, is some of the premises of our studies. So here's the lethality in the 154 knock-in mice compared to wild-type controls. And if we now look at RNA-seq data from the medulla and compare it to the cerebellum data, first at 18 weeks, we see there's a dramatic change in gene expression in the cerebellum, but essentially no detectable change in gene expression in medulla at 18 weeks. If we move to a later age, Not shown here is the cerebellar data. It's still high, but also dropping because now we're starting to see a loss of Purkinje cells. But at 28 weeks in medulla, we're now seeing changes in gene expression. And back studies indicates that we're starting to see, at least by RNA-seq, changes in the medulla, in the brainstem around 25 weeks of age in this model. Okay, I'm going to skip through this. I'm going to talk to you about that later if we want to. It's basically showing that there is some similarity in the gene changes between the medulla and the cerebellum, but the medulla does have distinctive features. Lastly, I want to close with some data from our ASO study that's being done with Ioannis and Holly Kordasevich. And we use a bolus injection into the lateral ventricle. It's very important to note that the ASOs fail to cross the blood-brain barrier. When we administer these ASOs to our SCA1 mice, and so these are ASOs targeting the mouse SCA1 RNA, we can see a positive effect on the motor phenotype, as I'll show you in a minute, lethality, as I show you, but the third feature of these mice, the weight loss, the failure to thrive, that is not corrected by ASO administration, which would suggest that that phenotype is driven by peripheral mechanisms as opposed to the previous two being CNS-mediated mechanisms. So here's the data on the rotarod. The bold are vehicle-treated 154s. The red are ASO administered, a single bolus at 5 weeks, and you can see improvement in the phenotype compared to age-matched controls. Here's the survival data. We get a roughly 7-week median increase in median survival, which is statistically significant with ASO administration, a single IV bolus at 5 weeks. And what we've just begun to do now is utilize these animals for cerebellar and medulla RNA-seq, trying to look for genes whose expression is corrected by ASO administration. And what you can see here is that in our RNA-seq data and some of the analysis that we've gone through, this is the basics of RNA-seq. We've gone through our first set of analysis that Christy has done is to look for genes that are significantly differentially expressed in the FEB vehicles versus the 154 vehicles, not different between FEB vehicle and 154 ASOs, and now significantly different between vehicle and ASO-treated 154s. We get 2 sets of genes, our cerebellar set with 158 genes, and our medulla set with 28 genes, no overlap. And analysis of the cerebellar set shows that there's a significant overlap with the magenta module, as well as this includes CCK. So a set of genes, if you will, that previous studies have been shown to be biologically relevant to disease. So that we're hopeful that this analysis is going to lead us to a similar sort of further understanding of what's going on in the brainstem, but that's yet to come. So basically, in the cerebellum and the Purkinje cells, the disease is due to the expression of the polyglutamine protein. It's largely due to alterations. The ATAXIA is due to Purkinje cell function, specifically signaling pathways, and the normal function of ATAXIN-1 is a coupler of transcription in RNA splicing. In the medulla, the RNA-seq data indicates that the disease begins much later than in Purkinje cells, and while there is some overlap in changes in pathways, there are some, I think, notable distinctions yet to be identified. This is the group that has participated in these studies, and those of you who know of our work, it's a long-standing 25-plus year collaboration with the Huda-Zogby group at Baylor. And I'm afraid I have to end on a very sad note. You've noticed that a substantial amount of the data that I talked about was generated by Jill. Unfortunately, Jill passed away about a month ago due to an accident incurred while biking in the morning in Minneapolis. One of the fortunate things about being a PI is the group you've had assembled around you. Excuse me. And the group has really rallied to keep her work going. And it's really terrific to see their dedication in moving her work ahead. I've gone way over time. So I guess we may not have time for questions, but I certainly want to thank you for showing up so early, perhaps after an evening of grappa also. And if there are any questions, I'm here for the rest of the meeting. And so please don't hesitate to ask me. Thank you very much.